Hello everyone, I am Anthony Bilodeau, a PhD student in the lab of Flavie Cardinal. I did my bachelor degree in physics engineering and my master degree in machine learning applied to the analysis of neuroscience images. Today I will be presenting how we can apply weakly supervised learning in biomedical imaging while still obtaining quantitative information. But first, I would like to introduce the challenges that we are currently facing and when we want to quantitatively analyze our datasets and the solutions that have been proposed. Over the years, the imaging techniques have evolved and can now produce tons and tons of data. For instance, Balsas et al. Reported data generation from a confocal microscope of approximately 5 megabytes per second, which produces images at a frame rate of 5 frames per second. If we scale that number over the course of a week, this can produce more than 200,000 images to analyze for the researchers. This number can increase to millions in the case of single plane imaging techniques. Researchers are also pushing the imaging techniques to acquire in a multi-dimensional fashion. For example, people have imaged plane or volume imaging over a time course, or also hyperspectral imaging, or also fluorescence lifetime imaging. The dimensionality of the data is a burden for the researchers who must analyze each frame of a video or analyze the hyperspectral cubes. Furthermore, eye-hand microscopes now combine multiple imaging modalities to correlate information, which again greatly increases the difficulty to analyze for researchers. People have combined single localization microscopy with stimulated imaging, depletion imaging, or also structured illumination with single localization microscopy. A solution that is proposed to analyze such data sets is, in most cases, deep learning. So as you all know, deep learning has now become the gold standard to extract information from raw, high-dimensional datasets. The possibilities to use GPUs in parallel computation on computational servers increases by many folds the speed of analysis. It has been used in many different cases in datasets to count, segment, or detect the structures of interest. Just so we're all on the same step here, I'm going to very briefly go through how convolutional networks extract information. If you'd like to precisely segment the penguins in the image using a DCNN, you would slide a convolutional kernel across the image and extract different feature maps. In this example, it is the edges of the penguins. To inform the network about its extraction of the features, we normally would use precise segmentation of the penguins as training samples. Those ground truth labels are then used to compare the segmentation of the network, which leads to a segmentation error, for instance, cross entropy. The error is then backpropagated through the network to update the weights of the convolutional kernels. As it can be seen, the feature extraction of the network becomes even better at extracting, for instance, the edges of the images. By applying a hard threshold on the image, one can obtain a segmentation from the network that is precise. While the usage of precise label provide a precise segmentation, it is longer to manually obtain the annotation in the case of penguin segmentation, we can think of using bounding boxes to facilitate the annotation period. However, in this case, we expect the network to be less precise, but still provide a general form of the penguin, since the network has seen multiple instances of different penguins. From this example, if we apply a hard threshold, we can obtain a segmentation that is less precise, but still better than the bounding boxes it was trained with. Now the question is whether we can apply weakly supervised deep learning techniques to obtain quantitative information about a dataset in biomedical imaging. And if so, How can we do that, and what types of labels can be used? So researchers in the field of biomedical imaging have already applied fully supervised deep learning techniques. For instance, to count the numbers of cells in the field of view, an annotator would simply put markers on the cells and then train the network to predict the position of these markers. To segment the cells, the annotator would precisely label the structures of interest and then train the network to output this segmentation map. A network that is used in many cases in biomedical imaging is UNET, as you probably all know. Recently, Falk et al. Reported their UNET implementation that could solve many common tasks such as detection, counting, and segmentation, both in 2 and 3 dimensions. However, one drawback of fully supervised techniques is that they need a precisely labeled dataset. To circumvent this problem, weakly supervised techniques were also applied to the field of biomedical imaging, which still provide quantitative information. Dubois et al. Used a unit-like architecture, but instead of using segmentation maps as a ground truth, they tried to predict the number of instances to segment in the field of view. Similarly, Cross et al. In 2016 used multiple instance learning scheme where an image is considered positive to a class if it contains the structure of interest. Using pooling layers, they were able to extract spatial information using solely binary labels. Finally, if you are interested in weakly supervised techniques, I would recommend that you read the review by Gina et al. From 2019, which reference a lot of architectures that were used in weakly supervised scheme. In Flavie Lavoie-Cardinal's laboratory, we are interested both in the in silico and the in vivo neural network. More specifically, we are interested at investigating the plasticity and growth of neurons. In other words, we are studying the formation of memory and how the brain learns at the nanoscale. The plasticity and growth of neurons is sustained by a cytoskeleton that is composed of numerous proteins, one of which is F-actin. We are interested at how the F-actin cytoskeleton is modulated and remodeled with activity in dendrites. A turning point of fluorescent microscopy imaging was the discovery of super-resolution techniques termed nanoscopy. Nanoscopy has allowed neuroscientists to investigate at the nanoscale the organization and localization of neuronal proteins. These super-resolution techniques has allowed the discovery of the F-actin particle lattice in axons that was later imaged in dendrites, but to a lesser extent. We have studied in the lab the F-actin particle lattice using super-resolution STED microscopy. While imaging F-actin, we could see 2 different patterns. At low activity, the protein F-actin forms a particle lattice, while at high activity, it would seem that the particle lattice breaks apart to form longitudinal fibers that are parallel to the dendritic shaft. To quantify this qualitative assessment, we sought multiple ways. However, we did not want to analyze the complete dataset by hand, since it is a long and tedious process. And furthermore, the dataset is always increasing in size since we repeat each experiment multiple times with different stimulations. For this reason, we tried using deep learning on our dataset. One way would be to train the network to use precise label as it is shown here for the fibers. However, this is a very long process that can take up to 3 minutes per image, which would result in a labeling time of more than 250 hours solely for training. To alleviate the task, we also thought about using polygonal bounding boxes, which still give spatial information, but reduces the labeling time by approximately 3 folds. Finally, another way would be to simply use binary labels of the images. For instance, on the image on the left, it contains fibers, while the image on the right contains rings or a periodical lattice. Using binary label would be very useful to reduce the inter-expert variability. So now that I have introduced weekly supervised learning, I would like us to go into the notebook. The goal of this first part in the notebook would be to first show qualitative performance of the network, of the trained network, but also show quantitative performance of the prediction of the network on their respective trained labels. All right, so this is the first part of the notebook, and in this part we are going to introduce weekly supervised learning. At the end of this part, you should be able to qualitatively visualize the performance of the trained networks, but also obtain a quantitative measurement of the performance of the network. So briefly, we are going to use a dataset that is composed of affecting dataset, that is composed of axons. So the affecting periodical lattice is present on axons, but the fibers are not present. So we are only going to predict the segmentation of affecting periodical lattice on axons. So this is a one-class problem. So at first, we can load some Python libraries here that will be used later on. Then you can simply load the data. Note that at the time of the recording, the GitHub has not been made public, but it will be made public for the presentation. So I have already loaded the data into the Collab notebook. Then we can load some user-defined utilitaries function that will be used later on. Now we can load the network information and the network parameters. We can see here that we'll be loading a polygonal bounding box model. So this model uses polygonal bounding box as a training labels. So the polygonal bounding boxes are the labels that I showed you in the presentation, while the bounding box model is the bounding box of the polygonal bounding boxes. So this will be clear in a few cells. So we can load this cell here. Then we'll have a look at the training curves. So there are two training curves. The first one here is for the polygonal bounding boxes, and this is for the bounding box model. What we can see from those training curves is that there is a black dashed line here, which specifies the model, the epoch of the model that that performed the best on the validation dataset. So this means that we are using early stopping. Then we can load the model here. In this case, we are going to load the polygonal bounding box model. So this takes a few seconds to load. So the next step is to infer the network on the testing dataset. So I wrote some utilitarian function here called showRandom. So this samples 5 images from the batch that is being inferred and shows it to you. So what we can see here, on the left here, this is the image that had to be predicted by the network. On the center column here, this is the labels that the network had to predict, and on the right here, it is a label that the network predicted. So this is a prediction of the network. So what we can see in some cases is that the prediction of the network seemed to be a little bit more precise than the bounding box annotation. So it follows better the shape of the axons. We can do the same for the bounding box model. Oh, sorry. So we can load it in memory, and then we can again infer on the testing dataset. Again, on the left here, this is the image that the network needed to predict, to infer on. On the center column, this is the label that the network had to predict. And on the right column here, this is the label that the network predicted. So what we can see is that the network didn't predict the bounding box annotation, but something in between a precise segmentation and the bounding box annotation. So now we can jump into quantitative assessment. So we'll use different metrics. For instance, the dice coefficient, the intersection over union. We'll also compute the confusion matrices, the precision, the recall, and the accuracy. So we can simply run this cell here, and remember that the model here uses the model that is currently loaded into memory. In this case, this is a bounding box. So this takes a few seconds to load. Sorry. So now the network has predicted all images into the testing dataset, and we can simply use these utilitaries function to show the scores. Okay, so we can see the dice coefficient, the intersection over union, precision, recall, and the accuracy. So we have an intersection over union of approximately 0.41. So remember that we are comparing the prediction of our model with the bounding boxes, which are very big. It is also worth noting that the accuracy is very high, of approximately 0.85. And this is expected since the accuracy is calculated with the background pixels, so the negative pixel class. And this negative pixel class is much more present in our images. So if we just go back a little to these images, so all those black pixels here are very more present in the images. So this increases the accuracy metrics by a lot. So this is the cumulative frequency of these distribution on top here. This is simply another way of showing those results. And here we have the confusion matrices. Now we can wonder how would a foreground detection impact on the calculated metrics. So for that, we'll use this utilitarian function here, which is called get foreground from the image. So we can run this cell here. Again, this takes a few seconds to load to infer on all the testing images, and we can now compare the results with foreground detection and without foreground detection. So on the left here, we have the performance of the network without foreground detection, and here with foreground detection. And what we can see is that the intersection over union increases a little bit, but that the accuracy drops by approximately 0.1. And this is, again, expected. So when we use the foreground detection, we are only looking at the pixels on the foreground. So the negative pixels, the number of negative pixels on the foreground and the number of positive pixels on the foreground is much more balanced. So this rebalances the metric. So this is a more this is a less biased metric for the accuracy. We can also compare the confusion matrices between without foreground detection and with foreground detection. Note that we can also do the same for polygonal bounding box. So here we can simply change polygonal, here, simply load the model again by running this cell, and this model is now loaded. We can go back down here and run this quantitative assessment. So again, this takes a few seconds to run through all testing images. So once this is done, we can show the quantitative scores between the two, between our labels and the labels of the network. And these are the scores that we obtain. So this is the confusion matrix. We can do the same thing as before. So by requiring the metrics to be only calculated on the foreground only on the foreground. And finally, we can compare the results before and after foreground detection. And what we can see, so there is no big difference between the two, the four metrics here for precision recall, intersection over union, and dice coefficient, but there is a small drop in accuracy. And again, this is explained by what I mentioned earlier. So the class imbalance between positive and negative pixels. So if we come back to the effect empirical lattice in dendrites, we can see that unfortunately not all images have structures that are well defined. In most cases, the images look just like here. As one can see, there are multiple distractors in the image. For instance, there is the presence of axons here and here, but also the presence of undefined effect in patches, just like here and here. As previously mentioned, we couldn't have used precise segmentation of effectin since it would have taken way too long. This is why we thought we could use polygonal bounding boxes to label all of our images in our dataset for both effectin rings and effectin fibers. One thing that is worth noting also is that in some regions of space, effectin rings and fibers can overlap, which again increases the difficulty to label for the expert. So if you remember correctly, before I showed you those two images, the image on the left was acquired at low activity, while the image on the right was acquired at high activity. Using a deep neural network trained on polygonal bounding boxes, we could show that there was indeed a conformational change from effectin rings to effectin fibers while increasing the activity. We could analyze more than 600 images, which saved us a total of approximately 300 hours of annotation time. So now I would like to go back into the notebook for the second part to analyze the real performance of the network. So as we saw in the first part, the prediction seemed to be qualitatively better than the labels the network was provided with. So our goal would be to assess the performance of labels that are better than the labels provided. To do so, I re-annotated some of the testing dataset and to show a performance increase with manual annotations, And also, I would like to briefly talk about some metrics. So not all metrics are well suited in biomedical imaging, and that some metrics may be more informative for a specific dataset. All right, welcome to the second part of the notebook. If you remember correctly, in the first part, we saw that there seemed to be a qualitative improvement in the performance of the segmentation of the network compared to the labels that it was provided with during training. And to do so, I re-annotated manually and precisely 30 images in the testing dataset so that we can obtain a quantitative measurement of the improvement, if there is one. Then I'm going to show that there is the possibility to craft new metrics that are specific to a dataset that may be more representative or more informative about the task at hand. In this case, this is the segmentation of the effect and pattern. Then I'm going to show that you can artificially increase the performance of the network by changing the threshold that is used in the output layer of the network. And finally, I'm going to talk about some a specific problem that can happen when we use intersection over union in comparing segmentation tasks. All right, so we're going to start by comparing the prediction of the polygonal bounding box with manual annotation. So while this is running, we are going to compare the polygonal bounding boxes with the manual annotations. So this is the expected value of the metric that we expect to have, since this is the labels that the network was provided with. Then we also compare the prediction with the manual annotations. So if the network has gone further than the labels it was provided with, we should see, in theory, an increase in performance. And finally, I'm also going to compare the prediction with the bounding box annotations. So this is what we saw in the first part. So I think this has run here. We can see here that this is some examples of some example images. This is the labels of the original labels, polygonal bounding box labels. This is the prediction of the network, and this is the labels that I re-annotated. If we compare the scores, we can see that there is indeed a small but noticeable increase in the segmentation performance. So when we compare the prediction with manual and the bounding box manual, we see that the prediction has gone further than the labels that it was provided with. Also, we can see that there is a clear increase in the performance when we compare the prediction with the manual annotations instead of comparing with the bounding box, polygonal bounding box annotations. You can do the same, but this time by using the bounding box model, and we should see a similar increase. Again, this is the labels that it was provided with, this is the segmentation of the network, and this is the annotation that I provided when I re-annotated the dataset. And we can again plot the scores and see that there is a small increase. So the network has gone further than the labels that it was provided with, since it performs a little bit better than the bounding boxes on manual labels. And however, what we see is that the prediction on bounding box is better than the prediction on manual labels. So the network is not as precise as manual labels, but it's still more precise than the bounding box on the manual labels. Another way to show this would be to craft a more representative and more informative metric. So in our case, we are imaging effectin, which is a particle. This means that in the Fourier spectrum, we should see an increase at the given distance between each actin ring. So if we were to compare the Fourier spectrum of the segmentation of the expert and the segmentation of the network, it should be very similar into this ring, so at this given frequency. While if we had a different segmentation than the expert, the difference should be higher. So in this case, a metric that is smaller, so smaller means that the prediction of the network is similar to the expert, while if we have a bigger difference, this means that there is that the network predicted a very different segmentation than the expert. So we can run this cell here to activate the implementation of this metric, and we can use the polygonal bounding box model. We can simply run now this cell here. Again, we can see some examples. So image, original label, prediction of the network, and my manual segmentation. And then we can plot the scores and see that there is indeed a smaller difference between the segmentation of the network compared to the manual segmentation than the bounding box than with the prediction bounding boxes. We could also try to maybe increase the precision of the network, of the bounding box network, by providing a different threshold. So to do so, we would simply re-annotate some images in the validation dataset using more precise segmentation. In this case, we are going to use the polygonal bounding boxes as a more precise annotation. So we can calculate the precision recall curve that we would obtain from the network to calculate an optimal threshold. So we can calculate this using this threshold here. And what we see is that the calculated threshold for the first 25 images seem to be 0.69. So we increased the threshold so that the labels become a little bit more precise. It is worth noting that the number of validation images here can also be varied. So if you want to relabel 10 images, then you can calculate a new threshold, or if you want to put more images in the, if you want to reannotate more images, you can also do to have a better representation of the threshold that you should use, more representative threshold. So with this new calculated threshold, we can then compare the scores of the models that are the original model and the model with an increased threshold. So this will take some time to run, but it should be done in a few seconds. And yeah, so now it is done. Again, we have the images that we provide the network with, the labels that the network was trained on, so the bounding box labels, the prediction of the network, and the prediction of the network when it is thresholded with a higher value. We can compare now the scores, and what we see is that there is an increase in the precision using between the prediction compared on polygonal bounding boxes and the prediction thresholded to a higher value with bounding boxes. And this again is similar for the precision that we could obtain when we compared with bounding box, so the original labels. One problem that can be faced with the intersection over union is when we are assessing small segmentation. And to show you this problem, we created a small example here. So we created a field of view of 20 by 20 pixels, and we put a ground truth circle of with a radius of 5 pixels in the center of the field of view. And then what we did is we created a prediction that is still a circle, but with a radius of 5 pixels. We also created a prediction, also again centered, but with a radius of 6 pixels. And finally, we created a prediction that has the good shape, but that is simply shifted by 1 pixel. So if we run the cell here, we can see the intersection over union that are calculated. So when we compare the ground truth labels with these labeled here, visually we can say that these are very, very, very similar segmentation. But the intersection over union tells us that this is quite dissimilar to the ground truth labels. And when we have the same area of pixels that are annotated but simply shifted by 1 pixel, we can see that the intersection over union is only 0.77. So now my goal will be to briefly mention some difficulties and challenges that will have to be faced in the years to come, and maybe start a conversation. As I briefly mentioned, some metrics that were created for segmentation assessment are not all well suited to assess the performance of a learning technique. The most shocking is, without a doubt, the intersection over union, which is greatly influenced by the size in terms of numbers of pixels of the structures to segment. However, the structures that we segment in my lab can be very small compared to the field of view, as you can see if we can see in the image right here. Moreover, the structures are not so well defined on the edges, which greatly impacts the scores of some metrics. In this specific case, an expert could be more strict on the measured signal and could label this image using these annotations, while another expert could label the same image using this type of annotation. In both cases, the segmentation is valid, but the metrics will be greatly influenced by the expert with which we compare our method. Another point that is worth mentioning is the difference in metric performance between natural images and biomedical images. Here, I reported the state-of-the-art mean intersection over union over all classes of the Pascal VOC 2012 challenge dataset of 0.89. In comparison, I also extracted the intersection over union of the state-of-the-art on the brain tumor segmentation challenge with the 2D architecture. As we can see, the performance on the old tumor is comparable to what is seen in natural images. However, one can see that by requiring a more precise tumor segmentation, such as with the tumor core or the enhanced tumor, we can see a drop of 0.1 and 0.2 in intersection over union. This shows the impact of smaller instances on segmentation metrics. It is also worth mentioning that, as I introduced in the network, in the notebooks, in some cases, a handcrafted metric by the expert can provide a more informative score. So if you're interested in metrics used in biomedical images, I would highly suggest the work by Taha and Anbury from 2019. Another challenge that I briefly mentioned on the previous slide is the expert variability. For example, we asked different experts to label effectin patterns using polygon and bounding boxes. As shown here, it is striking to see that the interpretation of effectin periodical lattice varies a lot from different experts. Mazara et al. In 2004 quantified the inter-expert variability on brain tumor. They reported a variability of approximately 30% between physicians, while the intra-expert variability was reported to 20%. Another challenge that must be acknowledged is the fact that the data sets between natural images and biomedical images varies in many ways. But the most striking difference is the number of annotated images. For example, in the well-known ImageNet dataset, there are more than 14 million images that have been annotated with a class, and approximately 1 million of those images contain bounding box annotations. The Pascal VOC 2012 challenge contains more than 11,000 images, with 7,000 of them being precisely semantically segmented. Cityscapes also contains more than 5,000 images that are precisely annotated. On the other side, in biomedical imaging, the cell tracking challenge contains 841 2D images that were annotated by trained experts. The Broad Institute also combines 25 datasets with 60 images per dataset as a median value. And finally, the Brain Tumor Segmentation Challenge regroups 243 images that were labeled by multiple physicians. So the difference in number is notable, and in most cases, it results from the fact that biomedical images requires a trained expert to carefully annotate the structures of interest in the images. Also, the images are acquired from biological samples, which are expensive to obtain, or from even human patients. So this lack of big data set leads to the generalization properties that are unknown for the training networks. For example, if you train a network on this type of sample with this type of noise, will this network perform the same on the same type of sample but acquired on a different microscope that has a different intrinsic noise? That is a question that has to be kept in mind. So along the presentation, we have proposed weekly supervised learning as a way to alleviate the annotation process and still provide quantitative information. I have also shown in the notebook that weekly supervised learning techniques can go further than the labels the network was provided with. Finally, I've also proposed that reducing the amount of information given to the network would prove useful to reduce the inter-expert variability. The question that you are now left with is, how will weekly supervised techniques help alleviate your research project? With that being said, I would like to thank the people that I work with in the lab. I would also like to thank our funding agencies and would like to thank you all for your attention.